Hello, my name is Aidan Bender, and I'll be presenting on biodiversity below zero, eukaryotic phytoplankton of the Arctic Ocean. Outside of the cute and terrifying polar bears that live up there, why would we want to focus on the Arctic Ocean? The Arctic represents half of Earth's polar ecosystems, with complex, unique, and extreme environments. It's also been shown to have heightened sensitivity to anthropogenic climate change, including panarctic increases in temperature and changes in ice melt and sea ice coverage patterns documented throughout the recent years. These changes ultimately cascade throughout the Arctic food web, affecting both the animals and the communities that rely on them in the area. Climate change has produced ripple effects for Inuit livelihoods, local economies, food systems, and general well-being. This project will be focusing on a portion of the base of the food web, protists. Protists are responsible for around half of the planet's primary production, as well as a myriad of other biogeochemical processes. They represent an essential trophic transition level between micro and macrofaunal, and their global ubiquity allows them to thrive in the Arctic's extreme environments. So what even is a protist? Protists are unicellular eukaryotes that possess an enclosed nucleus and are typically characterized by movement style and their specialized organelles. Historically, they're a bit of a catch-all group for unicellular microorganisms, with their diversity holding plant-like protists that can generate their own energy, as well as animal-like and fungi-like protists. Into our specific methods that we employed throughout this project, we refined eukaryotic-focused methods for sampling, DNA extraction, and DNA sequencing. This is in the goal to describe microbial diversity and construct novel protist reference genomes from the data. Ultimately, we hope to address gaps and form a comprehensive Arctic microbial baseline, where bacteria and archaea have already been analyzed. Eukaryotes haven't really been focused on in this environment. All of our samples, past and present, come from different regions of Inuit Nunangat. These are the land, water, and ice comprising Inuit homelands in Canada, and this project will be using samples collected from two regions, Nunatsiavut and Nunavut. These samples were collected along the Labrador Sea and into Davis Strait and the Baffin Bay area. In order to isolate eukaryotes, we employed sequential filtration, where we collected 2 liters of seawater from different depths and used a 20 micron pre-filter to remove any larger zooplankton and preserved a 3 micron filter that will limit any bacteria and viruses that are smaller than that, help limit the microbial noise to deal with in analysis. After the DNA extraction of these 3 micron filters, we are currently in the sequencing phase, where we're going to sequence the DNA and ultimately use bioinformatic analysis to discuss results. So how do we go about targeting eukaryotes? In order to do this, we look at an isolated 18S rRNA gene that encodes instructions necessary to synthesize the ribosome, which is responsible for creating proteins. Since this is integral to a cell's function, these rRNA gene sequences are highly conserved across eukaryotes, and we can pick them up across the DNA that we preserved on filters. Typically in literature, next generation sequencing has been used to get short primer regions around 400 base pair long to assess these gene regions. This project will be using both next generation sequencing with the short primers as well as third generation sequencing with long primers around 2000 base pairs long in order to maximize the amount of information that we're able to obtain from the DNA sequences. In order to identify the eukaryotes throughout the environment, we will compare variation between gene sequences against each other, as well as taxonomic databases, where similar sequences will be closely related and dissimilar sequences are distantly related, allowing us to identify who's in the environment and potentially what they're doing. Throughout the next steps of the project, after DNA extraction, we will continue to refine the methodology of amplification and sequencing. We'll use bioinformatics to analyze the sequencing, curating some high-quality assembled genomes, and compare the short and long read results, as well as we'll be able to assess the baseline diversity of eukaryotes, investigating what variables might impact community composition, such as depth, latitude, or geologic features. I want to thank you for listening to the presentation, as well as everyone that's made it possible to work on this research. And here are the references.